Hello friends, today we are going to discuss about one of the important topic in developmental biology about oogenesis and folliculogenesis, which is in continuation to yesterday's class. That means about spermatogenesis. Oogenesis and folliculogenesis. In previous class, we have discussed about spermatogenesis, which actually begins from puberty and continues up to old age, and the rate of sperm production is very high, say at the rate of 1.5 million of spermatozoa per minute. In case of oogenesis, it is nothing but the creation of ovum or egg from germ cells is different, is oogenesis. That means in contrast to spermatogenesis, oogenesis begins in the fetal stage itself. All the oocytes will produce during the fetal stage itself. That means unlike spermatogenesis, which starts from puberty, here oogenesis starts in the early fetal life. Since all the oocytes are produced in the fetal life, but many of them will degenerate. Around 2 million oocytes will remain at the time of birth. The most significant point is all these oocytes, they enter, that means they enter into meiosis, but they get arrested at prophase 1 of meiosis 1, what we call meiotic arrest. The rest of the stage is called dictate stage. That means just before the birth or after the birth, all these 2 million oocytes, much specifically primary oocytes, get into the meiosis 1, but they get arrested at prophase 1. That means at prophase 1 of meiosis, they get arrested. That's what we call meiotic arrest. And they remain in the arresting phase until puberty. We have discussed it is called dictate stage. And at puberty, they again start. That means they continue the meiosis. They become secondary oocyte, then ultimately egg. But it is not that simple. And we have discussed in yesterday's class now. For the primary spermatocyte, the secondary spermatocyte, the spermatid, ultimately spermatozoa. But here the similar concept but different. Here the thing is, the primary oocyte which actually arrested, that means just before or after the birth, in prophase 1 of meiosis, continues at the puberty. That means here it will finish off the meiosis 1 and become secondary oocyte. But the secondary oocyte which continues the meiosis 2, but again arrested at metaphase 2. This is very important. The secondary oocyte again get arrested at metaphase 2 of meiosis. When it will continue, just based on stimulation of sperm, when the sperm penetrates, it will continue the meiosis 2 and ultimately leads to egg, what we call oogenesis. Synthesis, creation of egg or ovum from the germ cells is oogenesis. Okay, just like spermatogonia in case of males, here oogonia is the egg mother cell. The sperm mother cell is spermatogonia, oogonia in case of here. But the most characteristic feature in oogenesis is the folliculogenesis. This is very important. Folliculogenesis are roughly spheroids in structures, and their cellular aggregations surrounding the oocytes present in ovary. They are just like fluid-filled sac-like structure which release hormones, and that means which in turn influences the menstrual cycle. Because we have discussed already, the primary oocyte at puberty becomes secondary oocyte, and secondary oocyte, because based on sperm penetration, become egg. What exactly this is? Menstrual cycle, puberty, the distinction between male and female is this menstrual cycle. In case of male, we don't have, that means the male person doesn't have any specific restriction or in production of sperms. Just 1.5 million sperm per minute, but here there is no such. Very, very complicated, and the quantity is also very less. Just one oocyte will enter into menstrual cycle. So we can expect just 500 oocytes in a female's life cycle. Here we are regarding follicles. That means we have discussed about, there are cellular aggregations surrounding the oocytes. They are just like nourishing or supporting cellular devices for the oocytes. Without these follicles, we cannot expect, normal processing of primary to secondary oocyte and so on. It means until menstrual cycle, like ovulation. Here, what exactly these follicles will they play? There are different kinds of follicles depending on the stage. Say primordial follicle, primary follicle, secondary follicle, tertiary or graphene or mature follicles, or atrial follicle. What exactly these? Depending on the stage. Say the primordial follicle will encircle the primary oocyte. They remain until the puberty, that means until the dictate stage. When the puberty comes, when they mature, this primordial follicle gradually becomes primary follicle. Then it enters into secondary, what we call growth follicle. Here we have discussed the primary oocyte should become secondary oocyte, and the secondary what will happen just before ovulation. What is ovulation? The release of egg. Particularly the release of secondary oocyte from the Graafian follicles is ovulation. It will happen once, that means there's one per menstrual cycle. For every month they will release one secondary oocyte from Graafian follicles, and it is ready, that means awaiting for the sperm. It's called ovulation. If the fertilization happens, then the Graafian follicle which is ruptured turns up into a corpus luteum, what we call a stereoideogenic cluster of cells. Stereoideogenic cluster of cells which have the ability to produce hormones. Which hormone it will produce? It will release progesterone. The corpus luteum will release progesterone, which prepares the endometrium of female uterus if at all the fertilization is success. That means success. Then that leads to implantation, pregnancy, and all that. It's about the basic overview of oogenesis and folliculogenesis. As we have discussed, a bit complicated when compared to spermatogenesis. Here, we can discuss about folliculogenesis. In which terms they are different with spermatogenesis? In nature and quantity. We have discussed primary oocyte arrest at prophase 1 until puberty. Then again secondary oocyte arrest at metaphase 2. Then based on that means waiting for the sperm penetration. And one egg, that means one secondary oocyte, simply called one oocyte or egg per one menstrual cycle. In general, this is about the distinguishing between spermatogenesis and the oogenesis. So the rest of the process is same, regarding fundamental issues like oogonia, the primary oocyte, what we call. But unlike spermatogenesis, it doesn't have in oogenesis, it doesn't have many subdivisions. Just oocytogenesis, okay. In general, first we'll discuss about oogenesis. Okay. We have discussed already in yesterday's class a primordial germ cell. A primordial germ cell which is extragonadal, born at somewhere. It will proliferate and that means it will migrate to the respective gonads. Say ovaries in case of females, testes in case of males. Then it becomes oogonia. Differentiates into oogonia. Then this oogonia become primary oocyte. The thing is, these primary oocytes are around 2 million at the time of birth, but they are under meiotic arrest. Meiotic arrest. That means prophase 1 of meiosis 1 until dictate stage. Dictate stage until they become attain the puberty. At puberty, it will continue the meiosis 1, then become secondary oocyte. Along with it, we will also have first polar body. Okay, this is diploid, and these are haploid. Then, and the secondary oocyte again under arrest at metaphase 2. Arrest. Then, based on sperm penetration, what we call induction, sperm penetration, it leads to that it will continue the meiosis 2, so that we can get egg and also second polar body. That haploid. So it's just an overview. Later on we'll discuss with respect to diagrammatic presentation. A simple flow chart how the oogenesis, just oogenesis, I didn't speak about folliculogenesis. We have to discuss integrated. Okay. First, the primary germ cell is extragonadal. Then it will reach out to the ovary, the oogonia, which is the egg mother cell, which is diploid, and it gives rise to primary oocyte, which is diploid. At the time of birth, it's around 2 million oocytes. And okay, before or after the time of birth, they start meiosis 1, but they arrest at prophase 1 until puberty, what we call dictate stage. Then when the puberty attains, the primary oocyte continues the meiosis 1 and becomes secondary oocyte, and first polar body. That means, but the unequal in size. Yes, first polar body is small inside, second oocyte is larger in size. Both are haploid. But the secondary oocyte, which has to continue the meiosis 2, starts but again stop at arrested metaphase 2. Then why? It has to wait for the sperm penetration for continuity. That means for completion of meiosis 2. When the sperm penetration is done, then the release of again two cells. One smaller one, second polar body, and the larger one, egg or ovum. Here the thing is, when the secondary oocyte is ready, it happens just before the ovulation. We have all discussed. Per each menstrual cycle, one oocyte will be released. With which stage? At secondary oocyte, and the follicle is Graafian follicle. We have discussed like this. Up till the secondary oocyte, the follicle, the name of the follicles we have to use are with respect to primary oocyte. The primordial follicle first, then the primary follicles, then the secondary follicles. Second otherwise called growth. With respect to secondary oocyte, we have to use that for Graafian follicles. Graafian follicle. Then it leads to ovulation. If it is succeeded, the ovulation is egg. The rupture of Graafian follicles will lead to corpus luteum. If fertilization is succeeded, it releases progesterone for fertilization process. That means for pregnancy ultimately. That means endometrium of uterus will be maintained by progesterone released by corpus luteum. If the fertilization is succeeded, then ultimately leads to pregnancy. It's a simple chart, but the thing is, the primary oocyte actually, all the females, say starting from a kid, which say a female kid until puberty, they contain primary oocyte arrested in prophase 1. But the thing is, initially they are surrounded by primordial follicles. But the primordial follicle significance is they remain dormant phase until 50 years. That's the significance. Since up to puberty, the primary oocyte will be covered by primordial follicles. Then when the puberty attains, the primordial follicle become primary follicles, then secondary or growth follicles. Then when the primary oocyte becomes secondary oocyte, it will be called the Graafian follicle. I told you now. We have to study integratedly. They are the cellular aggregations surrounding the oocytes. Depending on the stage, they may vary. Primary oocyte, the follicles corresponding to primary oocytes are primary follicles in general. When they attain puberty, they become primary, then secondary. When the primary oocyte continues meiosis 1 and becomes secondary oocyte, these secondary follicles, they transform into Graafian follicles. Then ovulation happens. Then the ovulation happens means egg, and these ruptured Graafian follicles become corpus luteum. If the fertilization succeeded, that means it actually depends on sperm, we already discussed. Sperm penetration leads to meiosis 2, then egg. Okay. If the sperm successfully fertilizes the egg, then the progesterone of corpus luteum will attain for a specific period. It prepares the endometrium of female embryo, that means female uterus, for pregnancy maintenance. So it's about the basic thing of oogenesis and fertilization. We have to study integratedly. Okay. Let us discuss about specific issues. It's about oogenesis and folliculogenesis. As we have discussed, depending on the kind of stage, we discuss about specific folliculogenesis. 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 Here, the first are primordial follicles. The second are primary follicles. The third are secondary follicles. The fourth are tertiary follicles. Tertiary are Graafian follicles, or mature, or antral follicles. Antral, cat-like. Secondary are growth. Just a basic representation. We'll draw about the particular diagram. Basic representation is like, for example, so that will be individually, then we can draw a representation. First, the primordial follicle. A primordial follicle consists of a flat squamous granulosa cells surrounding the oocyte. Flat squamous granulosa cells. We have discussed already. They remain dormant until 50 years. This is very important. When the puberty attains, this primordial follicle, actually which are flat squamous, they become primary follicles. That means at puberty. What is the distinction between the primordial and primary? This flat become cuboidal structures. Cuboidal structures. Then and all means the development of a layer called zona pellucida surrounding the oocyte. That means specifically, when the primary oocyte completes, that starts the meiosis one. That means at the puberty. When the primordial follicle become, that means during this process, primordial become primary follicle. And what are the distinguishing features? Here they become flat to cuboidal structures, cells. Okay. Then the zona pellucida is a glycoprotein polymer. It's a glycoprotein polymer surrounding the oocyte. Then and also genome activation means gene expression is on. Is very important. And also rudimentary paracrine signaling starts between the oocyte and the follicles. Signaling is most important. Signaling mechanism will start here. So when the primary, then at puberty, the primordial becomes primary based on this. So that means the development of glycoprotein polymer called zona pellucida surrounding the oocyte, and development of cuboidal cells, of course, and the genome actually starts. Expression is on. Then the signaling path. That means the rudimentary signaling starts between the follicles and the oocytes. Then further, secondary follicles. This is very important. Secondary follicles is a bit complicated. Complicated in the sense, till now just a single layer, but here they start. Actually, they undergo mitotic divisions. Mitotic divisions, so that we can expect around 9 layers of granulosa cells. Granulosa cells. These are all granulosa cells only, but they have different shape. Flat in primordial, cuboidal in primary. In secondary, it's like around 9 layers of granulosa cells. 9 layers of granulosa cells. At the same time, you can expect theca cells. Based on cytodifferentiation, you can expect theca externa and theca interna. And here the topmost layer of we have discussed 9 layers of granular cells. The topmost layer is called basal lamina. Basal lamina. So we have discussed secondary follicle, otherwise called growth follicle. Why? Because growth. Why it's called growth follicle? The word it implies. The follicle start undergo mitotic division, forming 9 layers of granular cells. Then the outer layers of granular cells is called basal lamina, and outer to it is they are called thecal cells, theca externa and theca interna. How they form? Actually one layer, but based on cytodifferentiation, what we call cytodifferentiation. They become theca externa and theca interna with intricate blood vessels between theca externa and theca interna. So it's like top layer theca externa, say it's expected secondary, theca externa, theca interna, granulosa cells, then granulosa cells, zona pellucida. This is oocyte. That means this is the division. Respect the secondary. Just I am presenting. Later we will draw a complete diagram. The thecal cells, the granulosa cells, the zona pellucida, and the oocyte. Such is the complication. That means growth follicles. Then when the primary oocyte undergoes secondary oocyte, that means complete meiosis 1 becomes secondary oocyte, and again start adolescent metaphase 2 just before the ovulation. Here, tertiary. The secondary follicle become tertiary. What is the distinction between the tertiary and the secondary? Development of antrum. A fluid filled structure adjacent to oocyte. The thing is, the same layers present in tertiary or graphene follicles or mature follicles, but in addition to that, an antrum will be developed. An antrum. So if it is oocyte, then this will be the antrum. This will be the antrum, okay? Antrum. And most important is, in tertiary follicles, the granular cells which are present in multi-layers, that means all they are undergo different sub-themes. Actually, on the whole, we have to say sub-theme in the sense, say corona radiata in graphene follicles. Corona radiata. Membrana granulosa. Membrana granulosa. Cumulus oophorus. Cumulus oophorus. Then periantral. Cumulus oophorus is called discus proligerus. The thing is, corona radiata present as surrounding the zona pellucida. It is all these terms and sub-themes we have to use when we are discussing about tertiary graphene follicles or mature follicles. That is the distinction with secondary follicle. Corona radiata surrounding the zona pellucida. We discuss now zona pellucida. Surrounding the zona pellucida, the corona radiata. Then membrana granulosa. Membrana granulosa means inward to the basal lamina. Just consider different granular cells in multi-layers. Now they become membrana granulosa. And connecting the corona radiata and the membrana granulosa, there will be a cumulus oophorus or discus proligerus connecting the corona radiata and membrana granulosa. We will discuss the antrum adjacent to the oocyte, and surrounding the antrum is the another layer, periantrum. So corona radiata 1, membrana granulosa 2, cumulus oophorus 3, and periantrum 4. So the 4 distinct divisions will be seen in graphene follicles, and most important, the antrum adjacent to oocyte. Just imagine, whenever there is a that means a second oocyte surrounds the graphene follicle is ready, means it is about to undergo ovulation, because secondary oocyte will be formed just before the ovulation. Just imagine, secondary oocyte formed just in ovulation means graphene follicle will form just before that, appropriately at this stage. Ovulation, here the secondary oocyte will release, that means from these mature follicles, and waiting for the sperm for the penetration. So they become egg, and these ruptured graphene follicle become corpus luteum. If the fertilization is done, it will prepare the pregnancy. That means it will release the progesterone. The progesterone will strengthen the endometrium of uterus for pregnancy. That means for implantation and all that. That means we have discussed already. Follicles and oocyte, we have to integrate the study, because if it is follicle, they surround, that means they are called cellular aggregation, simply in the form of fluid sac-like structure which release hormones and which influence the menstrual cycle. Through menstrual cycle channel only the secondary oocyte get mature. That is what we call the mainstreaming of oocytes to menstrual cycle, and done by, that means support or nourishment by the follicles. Appropriately, the different follicles in different stages. Okay. Let me come to the specific, presentation for better understanding. It's all about theory. Say this is oogenesis, this is folliculogenesis. First a diploid oogonia become a diploid primary oocyte. Diploid primary oocyte. Then this diploid primary oocyte undergo meiosis 1. Meiosis 1 to form first polar body, a small structure, okay? First polar body and a haploid secondary oocyte. It will undergo into meiosis 2. Meiosis 2 to form ultimately egg, a small second polar body. We discussed already primary oocyte. Now this primary oocyte and arrest at prophase 1. Second oocyte again arrest at metaphase 2. Then here the is egg means based on sperm penetration. Based on sperm penetration. Then this appropriate follicle now we have discussed already. Say for instance the primordial follicle. Primordial follicle. Then it becomes primary follicle. Primary. When primary follicle it becomes, and it starts from puberty. That means at puberty the primordial become, until the primordial will be in dormant stage, decade stage now. At puberty the primordial become primary. Then it becomes secondary. A bit larger in structure now. Secondary follicle. Then secondary oocyte now it become tertiary follicles. Tertiary follicle. Tertiary or Graafian follicle. Then after metaphyseal and what we call ovulation. Ovulation. They release the secondary oocyte outside. That means waiting for the sperm. Waiting for the sperm. This ruptured Graafian follicle become corpus luteum. The simple, that means in correlated way. Up till primary oocyte, that means at puberty state, it will become primordial follicle. When puberty start, the decade stage is end. It will, that means the primary oocyte which has been arrested at prophase 1 of meiosis 1, it will start and finish off the meiosis 1, become secondary oocyte. At this stage, that means before it forms secondary oocyte, it will call the first primordial become primary, the primary becomes secondary, what we call growth follicle. When the secondary oocyte is come, the follicle is called tertiary follicle. The four subdivisions: corona radiata, membrana granulosa, cumulus oophorus, periantral layer. Zona pellucida is formed in during primary follicle itself. Then ovulation in part 1 menstrual cycle. Menstrual cycle we have discussed already. Menstrual cycle. Then ovulation, menstrual cycle. One, that means the secondary will be released from the Graafian follicle and waiting for the sperm, so as to have to complete for the meiosis 2. That means to become egg. The ruptured Graafian follicle become corpus luteum. It will release the progesterone. It's about how the oogenesis and folliculogenesis will end. Okay. Let us draw a basic structure of Graafian follicle. This is very important because most of the structures are associated with that. Most of the functions, because hormones we have discussed already, released by it. Just because of lagging of time, just I'm dragging out a rough sketch. This is the secondary oocyte. Then this is zona pellucida. Then this is a corona radiata. Then these are cumulus oophorus. These are cumulus oophorus. These are cumulus oophorus. Then we have discussed already. An antrum layer will develop. These granulosa cells in multiple layers. Like this. There are granulosa cells. The granulosa cells. Then after basal lamina, there will be thecal externa, theca interna, and theca externa. Theca externa. Whereas these are intricate blood vessels. Intricate blood vessel between the two. Let me draw. This is the secondary oocyte. Secondary oocyte. Graafian follicle, Graafian follicle. Then this is zona pellucida. Zona pellucida. This is corona radiata. Corona radiata. This is cumulus oophorus. Cumulus oophorus. And this is membrana granulosa. Membrana granulosa. Then this is periantrum. Periantrum. And these are theca interna. And this is theca externa. Okay, this is antrum. You know, we told already. The significant feature of graphene protein is development of antrum adjacent to oocyte, second oocyte, you know. Then this is cumulus oophorus, then it comes the corona radiata with the top membrana granulosa. And corona radiata surround the zona pellucida. You know, membrana granulosa, the granulosa cells will transform into membrana granulosa. Outer is called basal lamina, then the red one is theca interna, the black one theca externa, integrate relationship. The thecal cells are most important, you know, they release estrogen hormone. They release estrogen hormones. Estrogen hormone.